The next speaker is Christine Iacobuzio-Donahue, and she did her MD at PhD at Boston University in the School of Medicine. She then moved to Johns Hopkins Hospital, where she conducted her residency, and then a postdoc, and then actually moved on to the faculty and became a full professor there. In 2014, she moved to Memorial Sloan Kettering Cancer Center, where she's an attending physician and also associate director of the Translational Research at the Pancreatic Cancer Research Center, and she conducts both clinical duties and research. And the title of her talk is Genetic Features of Subclonal Evolution in Pancreatic Cancer. Please welcome Christine.
First thing I'm going to do is take off my shoes. It's kind of something I always do when I give a talk. It's my signature. I so completely appreciate the opportunity to be at this symposium and to be able to be sandwiched in between Mike Stratton and Charlie Swanton. I feel a little bit like the middle of a basketball game when they bring like the 5-year-old out to try to get the three-pointer, kind of the entertainment in between the real shows. So I'm going to do my best, but really 98% of what I'll be talking about today is unpublished data. I really wanted to give an idea of what we're working towards, what we're most excited about, and just take advantage of this wonderful group. So this is Mike Stratton's talk that I don't... Yeah. Oh, it is on the screen. I see it there. Okay. Got it. Thank you. There's some things I'm good at and some things I'm not, so there you go. Okay, so yeah, we're talking about genetic features of subclonal evolution in pancreas cancer. There we go. Now I hit that button too quickly. Okay, here we go. So I just wanted to give a very quick overview of what we know about pancreatic cancer, how it forms, and the relative timing of when driver genes accumulate in the history of pancreatic cancer. So you see on the left side is the normal cells of the pancreas acini that produce digestive enzymes and normal ductal cells. That's my remote. Okay, because this one says Mac and that one says PC. Okay. And then from the normal cells that make up the pancreas arise pancreatic intraepithelial neoplasia, or PANN. So those are precursor lesions that occur in the ductal epithelium and the ductal system of the pancreas, and they undergo increasing grades of dysplasia and morphologic atypia spanning from PANN1, which is the first purple sort of bar, PANN2, and PANN3 is high-grade dysplasia. It's basically frank carcinoma in situ. Then, of course, you have an infiltrating cancer, dissemination, and colonization. The timing of the major driver genes in pancreatic cancer, there's many genes described, but KRAS, P16, P53, SMAD4, they occur during PANN. And they range anywhere from 95% for KRAS to 55% for SMAD4. And so these are really the big four genes in pancreas cancer. So this is a very recent publication from the International Cancer Genome Consortium, published in Nature, I think probably only a month ago or so. And this is... There we go. So here are the four that I just mentioned. So you see, even if you look at many, many tumors and you go deep and you're very comprehensive, these are the four that always come up at the top. These are really the major players. So those are the pancreatic cancer genes that we all think of. And then as you move down, there's, of course, chromatin modifiers consistently pop up. Their incidence often doesn't go above 30% or so of pancreatic cancers. For any given gene. There's a DNA repair cluster that continuously comes up, axon guidance, and then there are just other examples. So that is pancreatic cancer genetics as we know it, and in our field, we're pretty comfortable with those are the major players. So what we don't know is what is the extent of heterogeneity within a primary pancreatic cancer? Because all of these studies have always been done with a matched normal tumor, and if you look at multiple samples from the same pancreatic cancer, would you find that many of those other genes are actually there at much higher frequency? So that's one of the things we want to know. This is not advancing. There we go. What is the extent of heterogeneity of metastases in a single organ or different organs? Again, that's a big question. Are all the metastases derived from different subclones? Are they derived from one subclone that has equally disseminated throughout the body? So this is what we are striving to know. So I'll just, for a minute, step aside and let you know about the resource that we use. Which is basically tissues from patients who have consented pre-mortem to have an autopsy and allow us to take their tissues for studies. So this is a program that I started when I joined the faculty at Hopkins in 2002. I ran it for 12 years, moved it up to Sloan Kettering now. And we have done at Hopkins, it was 167 cases, most of which are pancreatic, and MSK, we're now doing all tumor types. And we do many things. For each person, for the primary, if it's present, and all of the metastases, and at least 10 different normal tissues, we collect tissue purely for diagnosis, confirm the diagnosis. We collect tissue that's snap frozen and it's processed for FFPE, for looking at biomarkers or anything else you want to do. We always attempt to immortalize some of the samples, either as xenografts or directly in culture, and there's an effort now to try to make organoids from these. And in the pre-mortem setting, so one of the things about this protocol is that patients can send pre-mortem, and that's the time when we collect blood for plasma for circulating tumor DNA studies and also for circulating tumor cells. So that is the material that we use. So in order to start to get at our big question, this is what we had to do. I'm showing on the right the key players. This is a wonderful collaboration with a lot of people, but the key players that really have done the work are Alvin McCahan-Moore, my graduate student, now postdoc, Johannes Reiter, who I see in the audience somewhere right in front of me. Hi, Johannes. And Ivana Bozic. I don't know if Ivana's here. So at the time we started the project, we had about 150 patients that were autopsied. We had to go through all of them. There were clinical exclusion criteria. So if somebody's pancreatic cancer did not arise from what we thought was a PANN, if it arose from an alternative variant precursor of a pancreatic infiltrating cancer, we excluded it because the genetics that go along with that are actually different. There's different genes. The patient had a history of a second malignancy. They were excluded. The patient was treated. They were excluded. That was the largest reason why patients were excluded from this study, because as you can imagine, treatment induces an exogenous bottleneck, and we don't want to induce any bottlenecks. We just want to know what is the natural history of the disease. And if they did not have any metastases at autopsy, this is actually a fascinating subset of patients. A side thing is not all pancreatic cancers are metastatic. But not appropriate for this study since we're interested in looking at metastasis. And then, of course, there's technical exclusion criteria, tissue quality, but one of the other reasons was that because many of our cases were coordinated outside of Hopkins and the tissue was sent to us, if the samples we had for that patient were not representative of their metastatic burden. So, for example, they might have had 100 metastases grossly evident, but we might have had 2. And so while the quality and everything was good, we didn't have the confidence at the time to look at everything. So that ended up with 4 patients. And I always go through this because if I just started out by saying we looked at 4 patients when we had 150, that it makes it look like we're maybe a little lazy. We didn't want to work that hard. We just picked 4. These are 4 very special patients. They all were untreated. We have their complete medical history. We have all of their tissue. It's a really unique opportunity to understand the natural history of pancreatic cancer. So this is what we do. So on the top left, you see this is an example of one of these tumors. They're large, they're bulky, they're heterogeneous just by eye. They can be different colors and consistencies, whatnot. I'm a board-certified pathologist, by the way, so this kind of stuff is, looking at that is interesting to me. We then take that tumor, freeze it, and then serially section it. So you see all the slices. So out of this one, we got 16 slices. And then, for example, I'm showing slice 9, where we score into 1 by 1 cm cubes best we can. And then each one of those cubes is bisected, always in the same orientation, the same way. And half goes into a tube for snap freezing, and the other half goes into a cassette for formalin fixation. So it's a beautiful method where you have whatever you sequence, if you find something of interest, you can go back to the tissue and look at it and see what are the features that go along in the morphology. If you're looking at the tumor and you say this is a really interesting or strange morphologic pattern, you have the tissue to know what happened in the tube and what was sequenced. So that's what we do. Here we go. So these are our 4 patients. We call them PAMs, PAM01 through PAM04. For PAM 2, 3, and 4, we had the entire primary tumor that was processed by multi-region sequencing. For PAM01, we did have samples of the primary tumor, but we did not have the whole thing. And the reason for that is PAM01 was one of the first cases I did before it occurred to me that heterogeneity was important. So once I realized that, then we started getting the whole tumor. So that was a missed opportunity. But we do have all the metastases for PAM01. And you see the distribution of these 4 patients is typical of what we see for pancreatic cancer. Liver metastases, lymph node, lung metastases, and peritoneal metastases. If you look at B, so what we did for the primary tumors of PAM 2, 3, and 4, even though we had the entire tumor blocked out and we had up to 50, 60 samples of each primary tumor, we picked 3 geographically distinct samples of the primary tumor, purposely making sure that they were as far apart from each other and just in an equidistant way. We didn't want to just take 3 samples right in a row from each other. And looking at region 1, region 2, and region 3, you see there's a little bit of morphologic heterogeneity just among these 3. So there's even a level of heterogeneity and complexity at that level. Very important point is in panel C, when we selected our metastases for these samples, we always made a point that we were taking metastases from different regions of the liver. And second, that the metastases were a discrete nodule. They were not sort of an area. Sometimes what happens with liver metastases or any metastases is you have many of them that are merging together. There might be tertiary metastases, things like that. We wanted to make sure this was a sphere and that there was nothing else around it. It was the discrete nodule, and we did that by doing serial sectioning and gross observation and everything. So these are individual spheres, and that also is going to be important to come up with. So what we did was basically, we had 39 samples from these 4 patients. That included the 4 normals, and then we had our samples of primary tumor, and then we had up to 8 metastases of each person. And we did whole genome sequencing at 60x, which, of course, generated a tremendous list of data that was filtered using the typical criteria you use to look for high-quality variants, and then every one of those variants was visualized by Alvin. It took him 6 months to just visualize that. And at the end of it, he still seemed to like me and was still enjoying his time in the lab. And so that ended up with a list of 2,579 high-quality somatic variants among these 39 samples, or I would say 35 samples for the 35 cancers. Then we go to the validation phase, which is targeted resequencing. So all of those mutations, we used a chip capture method to interrogate all of those to get much higher quality coverage. And the point of that was, of course, to validate the mutations that we thought by our filtering and visualization were high quality, to screen the additional samples we had for each patient, because we had a lot. And it's a very useful approach that we're able to determine the false positive and false negative rates, because any mutation that is in one patient, we had all of the samples from the 3 other patients as a negative control. It would be highly unlikely for the exact same point mutation, if it's a passenger, to occur in more than one person. Driver genes are different, of course. There's hotspots, but for a passenger, you wouldn't expect that. And this is just an example of all the samples that were sequenced by targeted and or whole genome, total of 181. And you can see, for example, for PAM 2, 3, and 4, that the number of samples sequenced was extraordinarily high, so it really gives us an opportunity to understand what's going on. So this is one way to look at the data, was hierarchical clustering. This is PAM02. A couple things to point out is we were very strict in our criteria in what we called what was present or absent. This is based both on filtering criteria for coverage and mutant allele frequency, but it was also based on a statistical algorithm to determine if a mutation was present or absent. So blue means present. We're very high confident that those are present. And as you can imagine, the blue ones are all of the driver genes, again. So that alone is a point. The question of the driver genes that you see, are we seeing heterogeneity? We did not. Number two is the reds are negative. Those are absent, and those are samples that we have very high confidence they're negative. The coverage was high enough, the quality was high enough, the cellularity, so we're very confident those are negative. Then anything in pink is unknown. The coverage was just not high enough to be confident that it was truly negative, okay? So we just do not count those. Oops. So another thing to notice by hierarchical clustering, and this is unsupervised or unweighted, is that you see liver metastases on the left are not clustering together in this case. They're kind of clustering within the primary tumors, and there's several instances where the liver metastasis is more similar to the primary than it is to another liver metastasis. So here is an alternative way of looking at the data by neighbor joining. I have the rainbow in the upper right to remind me to show you that if you look from top to bottom, it's in the rainbow scheme, and just makes it easier to remember that red is the most recent common ancestor, the parental clone of this person's tumor, and the pink all the way at the bottom, which is PT17, is actually the most divergent sample, the one that's at the end of this so-called subclonal evolution. This is not drawn to scale. You see most of the mutations accumulate from germline to that parental clone. So as Mike talked about just before, we refer to mutations are occurring throughout the lifetime. We're not just thinking of mutations in the tumor. So those 85 include the drivers, but it includes everything else that might have been in that lineage leading up to the finding of that parental clone. Notice primary tumor samples are called PTs, and liver samples, liver metastases, are LIMs. And so as you see, if you go across, that liver metastases seem to arise from different clones in the primary tumor. They're not all clustering together in this example. There is actually an element of subclonal evolution. The second thing that happened, I think one of the most important things that was found by Hannes and Ivana when they were trying to put this data together, is that it was impossible to generate trees without relaxing the criteria of persistence. You know, that a mutation, once it's present, it's never lost. It was impossible to generate a tree without relaxing that criteria. So you see, not only, when we often think of clonal evolution in tumors as mutations being gained, the reality is there's also mutations being lost, okay? So you have both gains and losses that go along. There we go. Did I skip a slide? No, I didn't. So there's another way of looking at it. So in order to give us confidence that this is actually working, we then, because we have the benefit of having the tumor mapped in 3 directions, is we go back and we take our rainbow scheme, so this is where that comes in and helpful, and we map it back to the 3D geography of the tumor. And you see that samples of different samples of colors cluster together. So that gives us confidence that our method is pretty good in determining these trees. It also shows, given the size of this tumor, about 5 by 7 centimeters, that the size of clones of the clonal fraction of maps that we are looking at are about 1 centimeter in size. You know, just to give you an idea of like what the size of a clone might be, and also that they're quite spread apart as well. So it's really quite amazing. Any sample that is in white, that's a cancer sample, but it did not have any mutations that were parsimony informative. And so therefore, they just are not included in this picture. Okay, so to get an idea, this concept of loss of mutations is very interesting in terms of what does that mean. So PAM01, this case had 12 loss events of 8 genes, okay? So to give you an idea that some genes are lost more than once, so we'll come back to that. This case, coincidentally, was also studied in a collaboration that actually my lab did with Mike Stratton at the Sanger, at looking at structural rearrangements in pancreatic cancer. And in this exact same tumor, so this was not coincidence, I'm always thinking ahead about what's gonna be a good thing to do. So this had 57 structural rearrangements, and we said, well, does that simply account for why you have losses? And there was actually no overlap at all with these mutations and the genes that were being lost. And we even had a very relaxed criteria. We went 1 million bases in either direction in that variant, and we still didn't capture anything. So it doesn't seem like, and we're working on other cases now, but it doesn't seem like structural variation can account for this. So then the next option is we looked at PAM02 as an example. This tumor had 51 loss events of 20 genes. 10 of those genes, or half of them, were lost 2 or more independent times. To see the range was up to 6, and for example, the mutations that were lost 6 times happened to be on the X chromosome. This was a female patient. So you want to wonder, well, what are these genes being lost? The genes by themselves, if you look at them, no particular pattern pops out as if, some exciting driver gene or something. But 2/3 of them were things that were acquired somewhere between the germline and the most recent common ancestor, or that parental clone. And then the other ones are acquired in a subclone and then lost in a later subclone. So it really points more towards an allelic loss, which we know happens during clonal evolution of tumors. Evidence in favor of that is several of the examples, the mutations that were lost at the same time. In a particular subclone, they were often on the same chromosome arm as another mutation on that same chromosome. So it kind of makes sense. If you lose 19p and there were 3 different mutations that were all lost at the same time, it really favors an allelic loss. Now, if the allelic loss is just random, if the allelic loss is trying to lose those genes and they have a function that we don't appreciate, or if it's a matter of there's something on that chromosome arm that it's being selected for loss because of haploinsufficiency of something else on there, I can't answer yet, but that's kind of where we are now, that we're really thinking these are allelic losses. The next thing we want to do is we want to understand what is the relatedness of these different samples. So essentially use the Jacquard index, which is looking at the fraction of mutations shared by 2 cells out of all the somatic mutations in either one of them, and the scale for a Jacquard index is 0, means there's no genetic relation between 2 cells, and 1 means they're genetically identical. So here are the Jacquard index values for the liver metastases of PAM02. And so this gets back to why we're looking at metastases specifically. You could say, why not look at the primary? Because the primary is we're looking at bulk tissue. There's a lot of mixing of clones or whatnot. With the metastases, since we know they arose from the primary, and we know that they were a sphere, and we're assuming that they were seeded by a single cell or a small cluster of cells, then we could feel confident that everything we see in the metastasis was that clonal fraction. And so you can see these are highly related to each other. These metastases that arose from different areas of the primary are highly genetically similar to each other. So you can also infer something about the primary. Now, in order to compare that, because it's one thing to say something is heterogeneous or not, but compared to what? So what we wanted to do was to come up with estimates of normal tissue, since single-cell sequencing is not at the level where we can be confident with the data, so we modeled it. Scenario 1 would be the zygote gives rise to stem cells during development, and then those become terminally differentiated cells. That's what you would see in a neural tissue like brain. Scenario 2, you develop self-renewing stem cells that are compartmentalized, and that is representative of GI tract epithelium. The pancreas also falls into this category based on developmental biology data. And then scenario 3 would be an example of hematopoietic tissue where the cells that are terminally differentiated are all mixed in with each other. And you see the predicted values by Jacquard for this does not go above 0.2. So So this gives us an idea of how cancers relate to normal tissues. So if you take a person and you take any two normal cells in an organ, in the liver, those two normal liver cells are going to be much more divergent from each other than two cells taken from the metastasis in the liver, okay? So the cancers are more homogeneous than the normal tissues, and that's just, as Mike said, things are accumulating mutations over the entire lifetime, so that needs to be taken into account. So I'm a pathologist, as I said, so once I see interesting patterns, I want to know what they look like. So here is PT1, the most recent common ancestor of this tumor, and this is what it looks like under the microscope: a conventional pancreas cancer, infiltrating glands, single cells, a lot of stroma, inflammatory cells. This is what you learn in medical school what pancreas cancer looks like. This is PT17, the most divergent sample in the exact same primary tumor, about 3 centimeters apart. You see you have a loss of stroma. There's a completely different morphology, and this is what it looks like at high power. One of the things I keep seeing over and over when I look at these cases is that subclonal evolution seems to correspond to a progressive loss of stroma, and I'm interpreting it as a gain of cellular autonomy, which kind of makes sense. You always say, what is it moving towards? You know, it doesn't have a conscience. It's not, you know, saying I need to get out of here and go to the liver. It's just changing over its environment. So there's this net gain of cell autonomy that seems to be the end game for pancreas cancer. And I'm just going to end on, because I'm also out of time, now that we know what to expect in untreated pancreas cancer, what do you see when you look at treated cancers, okay? So Zach Ahutek in my lab has taken this on. He's a radiation oncology resident slash postdoc. So while we're sequencing all the cases that we have right now, the best data we have is for stage 3 patients. These are patients that have pancreas cancer that is surgically unresectable, but they don't have metastases. So the paradigm for treating them is to give them chemoradiation. Every patient purposely, and we went through everything to make sure it was very clean, everybody had 50 gray radiation plus 5 FU. Everybody completed their treatment. And then at the end of treatment, people are imaged. And then some people, it turns out when they were imaged that first time, they had already developed metastases. Some people develop metastases much later. Some people only locally progressed, so their tumor got bigger, but they still didn't have metastases. Some of them didn't progress until quite late, right before they died. There we go. And then some patients had both metastatic disease and local progression. There we go. So since we said, and I said in the very beginning, when we looked at the untreated people, we had very strict criteria. Now that we felt like we knew what we were looking at, we're able to relax a little bit and look at all the other untreated cases that we had. I don't know why I am, there we go. This is for reference point PAM 1, 2, 3, and 4, okay? And then all the other ones we have. So these is what the trees look like at low power. Oops. And then this is treated pancreas cancer. And you see, just visually, they're more complex. The branches are longer. So now we have an idea of when you look at treated, what would you expect that the patient was never treated? And this is another thing when I ran in the lab this time on Tuesday, and I said, where is Zach? I need data immediately. Final slide. So how does this correlate to progression-free survival? We don't want to correlate to overall survival because somebody can progress and then still live quite a while. But in essence, it looks like, and these plots are kind of the inverse of what I showed before. The closer to 0 is the more genetically related. So you see are untreated on the left, and either looking at the mean Jacquard distance coefficient or the mean branch length, you see that the treated ones become much more diverse. And the patients that live longer have the greatest diversity. It's interesting, they still don't overlap the levels that you would see for normal tissue. Now, what does this mean? Do the patients, if they live longer, they have more chance to become diversified? if they are more diversified, do they have more metastases? They certainly don't necessarily become metastatic. Some of the most diverse tumors are ones that people lived a few years and never developed metastases. It gets back to that oligometastatic category I talked about. So that's where we are now. We're very excited. Number of things that we're really trying to chase down, and on that, I am done. This is my support, anybody in red who are the key players in this project, and I thank you for your attention.